Welcome to B005 Gene Expression. The central dogma of molecular biology states, DNA makes RNA makes protein. This two-step process, transcription and translation, is how the genetic information locked in DNA is converted into the functional molecules that run every cell. Understanding this process explains how identical DNA in every cell of your body can produce a liver cell, a neuron, and a skin cell, all with completely different functions. It is also the basis of all of molecular medicine, genetic engineering, and biotechnology. Let us go through it carefully. Here is something remarkable. Every single one of the 200+ cell types in your body carries exactly the same DNA sequence, the complete human genome. Yet a neuron extends long projections to transmit signals, a red blood cell is packed with hemoglobin and has no nucleus, a muscle cell is loaded with contractile proteins. How can the same instructions build such different things? The answer is differential gene expression. Different cell types switch different genes on and off using regulatory proteins, chemical signals, and physical packaging of DNA. The process of converting a gene into its protein product, transcription then translation, is the mechanism of that expression. Control this mechanism, and you control cell identity. This is why cancer biology, developmental biology, and gene therapy all focus intensely on transcription and translation regulation. Transcription is the synthesis of RNA from a DNA template. In eukaryotes, RNA Polymerase II is the enzyme responsible for mRNA synthesis. The process begins when transcription factors bind to the promoter region, a specific DNA sequence upstream of the gene. The TATA box, about 25 bases before the start site, is a key promoter element in eukaryotes. RNA polymerase reads the template strand in the 3' to 5' direction and synthesizes the complementary RNA strand in the 5' to 3' direction. Note that RNA uses uracil instead of thymine. After transcription, the pre-mRNA undergoes 3 critical processing steps in eukaryotes. A 7-methylguanosine cap is added at the 5' end to protect the mRNA. A poly-A tail of approximately 200 adenine nucleotides is added at the 3' end, and introns are spliced out by the spliceosome while exons are joined together. Translation is the synthesis of a polypeptide chain from the mRNA sequence. It occurs at ribosomes. Eukaryotic ribosomes are 80S, composed of a large 60S and small 40S subunit. Transfer RNA molecules are the adapters. Each tRNA carries a specific amino acid and has an anticodon that base pairs with the corresponding codon on the mRNA. Initiation begins when the small ribosomal subunit binds the mRNA at the 5' cap and scans until it finds the start codon August, which codes for methionine. The large subunit joins, and translation begins. During elongation, the ribosome moves along the mRNA in the 5' to 3' direction. New aminoacyl tRNAs enter the A site. The growing peptide chain forms a new peptide bond catalyzed by the peptidyl transferase activity of the ribosomal RNA, and the discharged tRNA exits through the E site. Termination occurs when a stop codon enters the A site, release factors bind, and the polypeptide is released. The genetic code is the correspondence between nucleotide triplets in mRNA, called codons, and the amino acids they specify. There are 64 possible codons, for bases cubed, but only 20 amino acids, so most amino acids are coded by multiple codons. This redundancy is called degeneracy. The code is universal, with minor exceptions in mitochondria. The same codon means the same amino acid in bacteria, plants, animals, and fungi. This universality is powerful evidence that all life shares a common ancestor. The start codon August not only begins translation but also codes for methionine, so all polypeptide chains begin with methionine, which may later be cleaved. The 3 stop codons UAA, UAG, UGA do not code for any amino acid, they signal termination. Crick's wobble hypothesis explains how a single tRNA can recognize multiple codons. The third position of the codon allows non-standard base pairing. The central dogma, DNA to RNA to protein. In some viruses including HIV, reverse transcriptase copies RNA back into DNA. This is reverse transcription, an exception to the standard flow. RNA polymerase reads the template strand 3' to 5' and synthesizes mRNA 5' to 3'. The mRNA sequence matches the non-template coding strand, with uracil replacing thymine. 3 key codons to memorize. Start is August, and the 3 stop codons are UAA, UAG, UGA. A useful mnemonic is you are amber, you are gone, you garbage. Ribosome sizes differ between prokaryotes and eukaryotes, 70s in bacteria, 50s plus 30s subunits, and 80s in eukaryotes, 60s plus 40s. This difference is exploited by antibiotics, streptomycin, tetracycline, and erythromycin target bacterial 70s ribosomes without harming human 80s ribosomes. Your NET strategy for gene expression. Codon and anticodon problems are a reliable source of marks. Given a DNA template sequence, derive the mRNA codon, then the tRNA anticodon. Remember, RNA uses U instead of T, and the anticodon is antiparallel and complementary to the codon. A classic in ET trap, Svedberg units S are not additive because they measure sedimentation rate, not mass. 60s plus 40s gives 80s, not 100s. 50s plus 30s gives 70s, not 80s. Memorize these combinations as separate facts. Also remember the eukaryote-specific mRNA processing steps, 5' cap, 3' poly-A tail, and splicing out of introns. Prokaryotes do none of this, they transcribe and translate simultaneously in the cytoplasm. These differences between prokaryotes and eukaryotes generate multiple NET questions every year. 5 key takeaways from B005. 1. The central dogma is DNA to mRNA to protein, with transcription happening first and translation second. 2. RNA polymerase reads the template strand 3' to 5' and makes mRNA 5' to 3' with U replacing T. 3. Eukaryotic mRNA is processed with a 5' cap, poly-A tail, and intron removal before it leaves the nucleus. 4. Translation begins at August, the ribosome moves 5' to 3' and terminates at stop codons UAA, UAG, or UGA. 5. The genetic code is universal, degenerate, non-overlapping, and commaless for essential properties. In B006, we apply all of this to genetics and inheritance, Mendel's laws and beyond. See you there.